Hey, it's Mr. Shrum, and welcome back to the physiology lesson walkthrough for the second section of the nervous system. We looked at impulse and how they function, the sodium-potassium gates of a neuron. Now we're going to look at impulse transmission. A nerve impulse, to review, is a wave of electrical current that spreads along dendrites or axons. Nerve impulses must be passed from one cell to another, or from one nerve cell to an effector organ or effector organ. And that can be muscles or glands. And the junction between one neuron and the next, or between a neuron and an effector cell, is called a synapse. Synapses act as one-way gates so that nerve impulses can only travel in one direction.
Synapses act as.
So here's a structure of a chemical synapse. You see how here's the dendrites and nucleus of one cell. You see the axon, and then you see the nucleus of another cell. And then if you zoom in really closely there, you see what's going on. And there are three components to a synapse: the presynaptic neuron, the postsynaptic neuron, and the synaptic cleft. The neuron that brings an impulse to a synapse is the presynaptic neuron. It ends in one or more knobs called synaptic endings. And the neuron that receives an impulse at a synapse is the postsynaptic neuron. So pre is the one bringing an impulse. The one receiving an impulse is postsynaptic. And the gap between the presynaptic neuron and the postsynaptic neuron is called the synaptic cleft. So that gap, we refer to that as a cleft. Transmission of the nerve impulse across a synaptic cleft or gap is a four-step process. 1. The action potential reaches the synaptic ending of the presynaptic neuron. 2. Calcium ions flow into the synaptic ending. 3. Synaptic vesicles release neurotransmitters molecules. 4. Neurotransmitters bind to specific receptor sites on the postsynaptic cell membrane. When a neuron is stimulated, there is a rapid change in polarity that moves along the nerve fiber, which is called the action potential. When the action potential reaches the synaptic ending of the presynaptic neuron, it causes voltage-gated calcium channels to open. Calcium ions flow into the synaptic ending. Calcium interacts with synaptic vesicles, resulting in them merging with the cell membrane. Synaptic vesicles release neurotransmitter molecules into the synaptic cleft as they merge with the cell membrane. And a good example of a neurotransmitter is acetylcholine, or ACH. Here, let's get another type of pronunciation.
Acetylcholine,
Yeah.
ACH.
Acetylcholine, choline. And it was the first neurotransmitter discovered. Once released, it diffuses across the synaptic cleft, and then it binds to specific receptor sites on the postsynaptic neuron. Neurotransmitter molecules have a different response depending on whether it is either an excitatory or inhibitory neurotransmitter. If it's excitatory, it acts to depolarize the membrane of the postsynaptic neuron. It does this by opening the gates of the sodium channels. positively charged sodium then flows into the cell, causing an action potential to continue down the postsynaptic neuron. So it's exciting the postsynaptic neuron. It's making things happen. Inhibitory neurotransmitter act to hyperpolarize the membrane of the postsynaptic neuron. And this happens by opening the gates of the chloride channels. Chloride ions are negatively charged and they flow into the cell. This increases the negativity within the cell, and this decreases the chances that the neuron will generate an action potential. So neurotransmitters excite a postsynaptic neuron by introducing positive sodium into the cell, causing an action potential. And inhibitory neurotransmitters introduce negative chloride ions, and that will reduce the chance of something happening with the neuron. Acetylcholine can be both an excitatory neurotransmitter or an inhibitory transmitter, depending on the type of specific receptor site on the postsynaptic neuron. After acetylcholine acts upon the postsynaptic neuron, it must be broken down to stop the stimulus and allow the postsynaptic neuron to receive new impulses. Acetylcholine esterase is, let's see.
Acetylcholinesterase, ACE,
Or sometimes referred to as ACHE, is an enzyme present on the membrane of the postsynaptic neuron. It will destroy acetylcholine after it has fulfilled its function. Transmission of the nerve impulse across a synaptic cleft is this four-step process. Action potential reaches the synaptic ending of the presynaptic neuron. Calcium ions flow into the synaptic ending. Synaptic vesicles release neurotransmitter molecules. Neurotransmitters bind to specific receptor sites on that postsynaptic cell membrane. And then depending on the type of the neurotransmitter, it will either continue the action potential down the postsynaptic neuron, or it will decrease the likelihood of the neuron generating an action potential in the postsynaptic neuron. So sometimes you want it to be excited and sometimes you don't want it to be excited. Yeah, but that's it for impulse transmission. If you have any more questions about this topic, message me in Canvas and I will work with you one-on-one. But until next time, have a good one, and I will see you all later. Bye-bye.